cold in here, isn't it? Okay, well, good morning everyone. Thanks to those of you that have made the trouble to come in today. What I'm doing in this lecture is quite different to what I've been teaching you about earlier in the course. But this is actually a topic that I've been teaching in the pharmacy program for some time, and realising that we need to develop it significantly there, because this area, the topic I'm talking about today, has a big impact on pharmacists. But it's something we need to talk about too in a course like this for pharmacology students, because this is a reality of our world, that the nature of the substances that we use to treat disease is changing significantly. And the problem is that pharmacology departments and pharmacology courses have tended to focus on small molecules, but in reality the practice of the current time, and certainly in the future, judging by current trends, is going to be more heavily weighted towards larger molecules. We sometimes call them biologics. So what I want to do in this lecture is just review this issue, and it's going to be very basic. There's not a lot of science in this lecture, it's just an introductory one, just to make you aware of this sort of change that's happening in medicine. And I want to introduce some of the basic scientific aspects surrounding this issue, but also some of the commercial and practical issues that accompany the use of large molecule biologics in modern medicine. Now, why has this revolution happened? Well, it's just really a predictable consequence of the way in which our understanding of biology and disease has sort of revolutionised just in the time that I've been in science. I guess I'm pretty old, but it just seems that in the last 30 years or so, we've gotten a lot better at understanding the molecular details underlying biological function in both healthy tissue, but also how it changes in disease. So, you know, when I was a boy, we really didn't learn anything at all about a lot of these topics: genomics, transcriptomics, how the expression of genes within tissues changes as a result of the onset of disease, how that results in changes in the abundance or declines in concentrations of particular proteins, then how that relates to changes in all the various hundreds and thousands of metabolites that are produced by enzymes and proteins, and then of course advances in computing and the area of bioinformatics have given us unprecedented tools for mapping all these complex changes in biological systems. Now, so the upshot of all this is that we now know a lot more about particular pathways and particular proteins that play roles in diseases. And in the days gone by in pharmacology, we would try to make small molecules to disrupt the activity of a protein or receptor that's getting out of control in a particular disease. We'd spend a lot of time designing a small molecule to block that protein or to stimulate the protein if it's deficient, and come up with a orally administered drug. But that takes a lot of time, and it's just a reality of science that a lot of the proteins that are changed in a particular disease that we might like to inhibit with a drug are actually pretty tough to inhibit selectively because they're not very druggable. the protein isn't amenable to modulation with a small molecule. Okay, so in this environment, one of the easier strategies has been just to develop an antibody, therapeutic antibody that takes out a particular protein and blocks its role in a pathogenesis of a particular disease. So this is because they're largely easier to make, although they're very expensive, and we'll talk about some of the implications of shifting towards this sort of large molecule-based therapeutic approach in disease. Okay, so this shift towards using large biologic therapies in the treatment of disease, particularly the use of monoclonal antibodies, really is having a significant impact on the clinical control and management of people with some pretty nasty chronic long-term diseases. And some areas where we're seeing particular impact would be things like rheumatoid arthritis, where these biologics really have revolutionised the treatment of more severe cases of this disease. Plaque psoriasis, also a nasty condition that can affect people over many years, that can be a chronic condition that's really quite disfiguring. Also is an area where we've seen a significant impact of biologics. They're also having an impact on life-threatening cancers as well. But there are many other areas of medicine where biologics are undergoing or eliciting a bit of a revolution in how we treat disease. Also, we can just see how the future is likely to look, given by the fact that currently about a third of all clinical trials are involving testing of biologics, large molecule interventions rather than small molecules. But this comes with major implications for societies, because these things are extremely expensive. So they can, for some of these biologics, can cost up to $1 million a year to treat a single patient. So where's that money going to come from? And that money is well invested if it results in significant improvements in downstream costs for caring for that patient. But if it only results in modest improvements, then it's really going to put a lot of financial stress on our healthcare. This is a challenging area to get your head around and to teach, because like we've seen in other areas of drug usage over the years, sometimes drugs can be known by different names in different countries, and this also is a challenge in getting your head around biologics. Now, I'm already using this term biologic without defining it, so we better make sure that we understand what we mean when we use this term. There are a couple of definitions that are offered in the literature. One is a medically active substance made by or derived from a biological source. So this is where it came from is what makes a molecule a biologic. So it's something that's derived, say, from a cell-based production process rather than a chemical synthetic one. Alternatively, a molecule whose composition mostly entails naturally occurring amino acid sugars or nucleotides also constitutes a biologic. So something like a antisense DNA, for example, or an mRNA vaccine would be considered largely a biologic under those sorts of definitions, even though they're made synthetically, but they try to reproduce a biological structure. Okay, so those two terms are often used in the literature. Note that other people use different types of names. Some people call them biopharmaceuticals, biotherapies, so different countries might prefer to use these sorts of terms rather than biologics. And also be aware of the way we refer to different generations of biologics. Remember, if we did drugs that changed the world, we talked about that issue for certain small molecule pharmaceuticals, like certain antibiotics, where we talk about first gen, second gen, third gen, fourth gen, based on when they originated. Same is true for biologics to some degree. But we also use the term innovator biologic, or even the originator biologic, just to refer to the first biologic that's the first member of the class of therapeutic agents. So the first TNF alpha, for example, trapper, would be called an innovator biologic. Now, if we look at the types of biologics that are currently in use, and this isn't completely detailed, but therapeutic proteins. Now, we've been using some of these for a long time in medicine. Insulin, for example, human growth hormone, have been around a while. In the old days, we used to extract some of these hormones from different animal tissues. But now we've obviously got recombinant technology capacity, so we can make human versions of in a biological system on a large scale. Blood clotting factors, antithrombotic factors also been around for a long time. Erythropoietin, been around for quite a while, used to stimulate red blood cell production. Interferons, growth factors, cytokines also have been available for some time. The one class that is really growing though are the so-called therapeutic antibodies. These target usually a single antigen, although now we're seeing bispecific antibodies that can target two antigens in a biological system. Then we come to therapeutic nucleic acids. Antisense nucleic acids are growing in popularity, increasing number of them on the market. The odd silencing RNA is also coming through. And then there are these new classes of biologics such as antibody drug conjugates and fusion proteins, and these are also emerging as an approach to a class of biologics. Now, I just want to spend a bit of time now thinking about why is this a big deal? Why do we need to even talk about this in a pharmacology course? Well, the reason we need to talk about it is probably, as David Joyce, our unfortunately departed, or at least retired clinical pharmacologist, used to say, if we don't teach it, no one else will. And it's a job of a pharmacology department is to help people think about the biological effects and fate of molecules used in the treatment of disease. So if we don't help people get their heads around biologics scientifically, then probably not many other disciplines will. But it is a challenge to teach biologics because they're just completely different in terms of their behaviour in the body to the sorts of molecules we normally have been focusing on in pharmacology departments for over 100 years. Okay, so if you think about small drugs then, that you've probably heard a lot about in pharmacology lectures, they still comprise about 90% of prescriptions. So we need to keep biologics in perspective. Most of the prescriptions written by doctors and filled by pharmacists are still for small molecules. And there's a number of reasons for that. The most straightforward reason is that you can get them into the body via the mouth and via the oral route. So it doesn't take any sophisticated technology to give the drug to the patient. Just tell them to take a glass of water or milk with their pills, and you can send them off and distribute the drugs via pharmacists easily and make it relatively easy to take these sorts of therapies. They also have a fairly simple structure in chemical terms. So they usually have masses of less than 500 or 600 grams per mole. And this has implications for synthesis and production of these molecules. It makes that relatively easy, and it's something that can be replicated when a drug goes off patent and we start making generics. It's relatively easy to reproduce the chemistry because organic chemists are experts at that. It's also relatively easy to formulate them for oral preparation, although that's more challenging for some drugs than others. But generally speaking, it is possible to formulate most effective small molecules for oral usage. They're also well distributed within the body. Go away. And often they have cell-penetrating properties and can access intracellular targets. Because they're small, they can get inside cells and produce a biological effect by targeting some intracellular process like a non-steroidal anti-inflammatory like aspirin or ibuprofen that blocks the cyclooxygenase located within the cell. So the drug can get through the membrane and access its target. But also, because it's a relatively small molecule, you're not relying on too many chemical forces during the interaction with its target receptor. So this raises the possibility of non-specific binding with other proteins. And so that means that the typical small molecule drug has side effect issues because they can alter the activity of other targets, not just their preferred target. Okay, so that's got implications in terms of adverse effects. They also have short plasma half-life because, as I've been emphasising in my lectures, they undergo usually quite rapid metabolism in the liver. They get transported into the urine out of the kidney by transporters. And so the body has a whole lot of biological processes for protecting it against small molecules, and pharmacologists have learned a lot about those systems by studying them for many, many decades. So we know a lot about them. And it also means that you need to take frequent doses. The fact that the drug is getting metabolised quickly, have to replenish the levels in the plasma for the patient by giving repeated doses. And as I've been highlighting in my lectures on biotransformation and transport, the fact that relatively few P450s and transporters protect the body against so many drugs means that there's a possibility of DDIs when you inappropriately combine drugs together that all depend on cytochrome P453A4 or all depend on P-glycoprotein for clearance. Means they can screw up each other's biotransformation and clearance. Also, of particular relevance to toxicologists, that problem of DILI I talked about earlier, drug-induced liver injury. Sometimes drugs get converted into nasty metabolites, small molecules that is, into nasty metabolites that start attacking the proteins or DNA and cause toxicity. Also, it's not perhaps obvious to people, but there's a lot of expertise in big pharma regarding how to make small molecules, how to overcome any deficiencies they might have during formulation, how to enhance their pharmacokinetic properties. All that sort of knowledge is pretty well established in-house within companies like Pfizer and Merck and those sorts of companies. So they've been doing this for many generations and have gotten very good at it. They have huge compound libraries. They know a lot of medicinal chemistry, how to tweak the structure of molecules and what's achievable. And as I said, it's also easy to make these molecules, and this means that when the drug goes off patent, then it's relatively easy to shift the production of the molecule offshore to a low-cost environment like China or India. And I think if you get a prescription these days, you'll see a lot of the medication supplied in Australia come from India. It's really a major centre of pharmaceutical production for drugs used in the Australian setting. So it's relatively easy to do that, and it's not that risky because it's pretty well established, all the protocols for producing a given drug, all the chemical steps involved and the industrial processes required to carry a molecule from the starting material through to the final pharmaceutical product. It's pretty easy to go and check to see that the company knows what they're doing and they're checking for contamination and problems that might occur during each stage in the synthesis of a product. Another issue with small molecules is that thankfully immune toxicity is relatively rare. There are some exceptions, and I've got to give you a lecture in a few weeks on drug-induced allergies in people that have inherited HLA variants. It's a big issue these days, but still it's pretty rare. We'll see that this isn't necessarily the case with large molecules. The bigger the molecule gets, the better the immune system is at detecting it and reacting against it. And so relating to that ease of synthesis, relatively easy to maintain good consistency from one batch to the next batch because it's a small molecule. Chemists are pretty good at their job, and we have lots of analytical technology for appropriately determining the presence of contaminants and byproducts of synthesis within a pharmaceutical batch, so we can confirm that the product the patient's getting is really pure and high quality. Also, there's not many issues with contamination usually. So because we're using organic solvents, there's not issues with bacteria getting established in your some component of your production laboratory or equipment. And also from a pharmacology point of view, we also know a lot about these molecules. We know a lot about their pharmacodynamics usually. We know a lot about their pharmacokinetics and how the structure of the molecule impacts both pharmacokinetics and pharmacodynamics. Okay, so that's all part of the reason why small molecules are so well established. Now, biologics are different on pretty well every one of those criteria. So they have highly complex structures. They often have molecular masses of in excess of 5,000 grams per mole, and now in the year of mRNA vaccines, they're blinking humongous things, 1.4 million Daltons, so much, much bigger than a little drug that's taken via the oral route. And because they're so huge, they don't get across biological barriers very well or get into cells, so we need to come up with novel delivery tools and methods for getting these things into the body. So usually we use injections of various kinds, subcutaneous or intramuscular, sometimes intravenous, to get past the biological barriers to the entrance of large molecules into the body. That comes with issues and complexity. Who's going to administer it to the patient? You can't rely necessarily on your poor old stressed out local pharmacist to do some pretty complex administration approaches with particular biologics that have issues. Another issue with these molecules is that they mainly hit extracellular receptors. So they don't have great tissue penetrating properties and cell penetrating properties. So if you're going to block inflammation, you need to block a extracellular mediator of inflammation like a cytokine. You can't interfere with the cyclooxygenase activity that produces, say, prostaglandins. But one of the great benefits of a biologic is that it has excellent target specificity, low non-specific binding. So you can just take out a particular protein that's role in disease and not worry too much about off-target toxicity and side effects. That really is a positive property of these molecules. Another property too is that because they are the mechanisms involved in the pharmacokinetics of biologics, and this is something that's really only beginning to be properly studied now. There was just a nice series of reviews in, I think it's one of the drug disposition journals just earlier this year, a series of reviews on biologics saying we really do need to know more about the pharmacokinetics of biologics, and one of the reasons why biologics may fail in clinical trials is that people haven't been thinking about pharmacokinetics of these molecules very much. So there's growing awareness that we do need to think about them more than we have been. But one of the great advantages is that with a lot of biologics, you only give it to the patient every month or so. They'll come in and have an injection, and that will, the molecule is sufficiently long-lived to stick around for a significant period of time. And this is partly because of the clearance mechanisms. Are often lymphatic in nature, which is lymph is really slow. Just small quantities of lymph get cleared of stuff every day, and so these molecules have really long half-lives rather than the rapid clearance mechanisms of the kidney or the liver for small molecules. And because they don't use drug metabolizing enzymes like P450 or transporters like PGP means, they're really free of all that drug-induced or drug interaction concern with small molecules, which is really a positive thing. But we have found in just recent years that they do have some issues. DDDIs, I don't think I talked about that in my lectures on drug metabolism, but DDDIs are disease drug-drug interactions. And this is when, we've seen this with some of the biologics used to treat rheumatoid arthritis. What we didn't realise is that the expression of P450 genes is under the control of inflammatory cytokines, and so when you trap that cytokine, you can change a person's cytochrome P450 activity, and this can affect their metabolism of small molecules. So this is an area that's growing in significance in clinical pharmacology. They also rarely form toxic metabolites, so that whole problem of DILI, drug-induced liver injury due to toxic metabolites, is pretty well gone. And then there are completely different companies that have emerged during the biotechnology era to cope with and focus on the development of large molecule therapies, like Amgen and other companies like that have been in the game for a while now, but completely different knowledge base than would traditionally be associated with a company like Pfizer. But one of the big issues, and I'll come back to this in a minute, is how do you make a copy of this molecule when patents expire? And if you want to produce a low-cost copy of a patent, of a expired molecule, by shifting it off to India or China, then how do you know that what they're making is identical to the product you're getting from the established company that originally developed the biologic? So that's a really big issue that governments are working through at the moment. Also, unlike the rarity of immune responses in people receiving small molecules, immune responses are very common with biologics. A high proportion of people after an infusion with a biologic will have a fever, high body temperature, sometimes rashes, that sort of thing, all due to your immune system reacting to the large molecule. And also batch variability becomes a huge problem, as does susceptibility to contamination and impurities, simply because we're dealing with biological production systems, cell culture, things just go wrong in cell culture, and it's very easy for some of the contaminants due to infection, a viral infection of a cell culture, for example, might cause contaminants of a biologic. It's very easy for that to happen. They also have complex actions, both in terms of pharmacokinetics and pharmacodynamics, and often we just don't know really how they work in a complex in vivo setting, although we like to think we do. But then at the top of the list here, these dollar signs show the implications of all this, that small molecule drugs are a lot cheaper to produce and use and deploy than biologics. So this is raising significant questions in healthcare, dealing with some of these issues. It's just a diagram of some of the newer classes of biologics that are coming through the system now. A fusion protein, and here the approach involves fusing a say an antibody, an IgG Fc domain, with a particular protein of interest through a sort of a linker, chemical linker. And this is a way of facilitating receptor-mediated uptake of some of these proteins. So this may allow us to go after intracellular proteins to a degree that we haven't previously with other biologics. And another approach too that's starting to come through is an antibody-drug conjugate. And this is a fascinating idea where we're chemically linking a toxic small molecule, such as an anti-cancer drug that's toxic to tumour cells. And the whole problem with using small molecule anti-cancer drugs, of course, is off-target toxicity in other organs. Your gastrointestinal tract, suffers a lot of damage, or your hair falls out, all those sorts of vomiting and diarrhoea, nausea, common side effects of some of these drugs. But by linking it to a protein that is engineered to interact with a antigen, with a tumour antigen on a tumour cell, and then undergo receptor-mediated uptake. This is a way to minimise off-target toxicity from some of these anti-cancer drugs and just ensure that they're producing their harm within a tumour cell rather than a healthy cell. So these are some interesting technologies. Now I put this diagram together just to show how this world does really challenge pharmacology ways of thinking about small molecules, because we've long been focusing on tiddlers like aspirin, tiny little molecules with a molecular mass, just 180 grams per mole. And then along came the first biologics, insulin, close to 6,000 grams per mole, erythropoietin, 30,000 grams per mole. And then we thought that monoclonal antibodies were monsters with molecular masses of 150,000 grams per mole. But now in the era of mRNA vaccines, so this is I couldn't find a picture of the mRNA molecule, but it's a huge molecule, 1.4 million daltons. So, and as I said, the issue with a small molecule is that you can access an intracellular target quite nicely because they act intracellularly, readily enter cells across the cell membrane. Whereas most biologics, once you start getting above 800, 600 grams per mole, then you can't access the cellular interior. So most of these, like insulin, interacts with an extracellular receptor. Monoclonal antibodies often trap cytokines and extracellular or their receptors. But the companies making mRNA vaccines try to persuade us that they can access intracellular targets just by lipid nanoparticle formulation with clever formulation technology, so you can get a monstrous molecule into a cell. Now that obviously raises a lot of unknowns about the toxicology of some of these products. In my opinion, they were probably rushed to market a bit too quickly, and there's some significant side effects there. But hopefully we'll make some progress in this area, and companies will be a little more forthcoming with their technologies than they have been in the COVID era. Now, so this revolution is happening, whether we like it or not as pharmacologists, and this is just a diagram showing the proportion of biologics making to newly approved drugs coming through the system in the United States. So this is FDA approved molecules from 1993. That's when I started my career as an academic, and you can see just a couple of the molecules that were approved in that year were biologics. But now, last year, 15 relative to 22 small molecule or new molecular entities. Out of the new molecular entities, about 38, I think, 15 of them were biologics. So that's a third or higher. Okay, so we break these down. What are these new products coming through? Well, this is from the 2022 FDA approvals. You can see small molecules, 18 of them were small molecules. And then we have these therapeutic proteins of various kinds, including monoclonal antibodies and bispecific antibodies, an antibody drug conjugate as well, and then a few other recombinant proteins and enzymes. So biologics are growing in market penetrance, and for the reasons I mentioned, that they do allow specificity in targeting particular proteins. So that's probably gives us new approaches in treating some of these diseases. Probably the most important enabling technology that facilitated this move to a more biologic-rich therapeutic environment would be the capability of making hybridoma cells, this hybridoma technology. And I think this was developed probably back in about the late 80s, not quite sure, but been around a while now, but it's gotten better and better and better at large-scale production of these things. But the thing with a hybridoma is that you can take a polyclonal, unlike a polyclonal antibody made in animals, monoclonals can be made in vitro. And you do it by fusing the B cells from the spleen of an animal with cancerous immortalised cells, myeloma cells, and this produces these hybridoma cells. So they can churn out, you can grow them in vats and churn out very specific antibodies. So this technology really did allow for scaling up of production of antibodies on a scale not previously seen. But it's still pretty costly to do this. So it takes, gallons and gallons of culture media to make enough monoclonal antibody for a single human dose. So that's one of the factors that makes this technology so expensive. And as we saw during the COVID era, when people found that some of these anti-cytokine biologics, monoclonal antibodies, were very good for treating COVID patients, produced a shortage for people that needed them for rheumatoid arthritis. So having the capacity to produce enough of these biologics to meet everyone's needs can be quite challenging in the pharmaceutical sector. Now, another technology that's happened as time has progressed is we've realised that relying on mouse antibodies comes with significant issues with regards to immune responses. And so as time has progressed, we've seen increasing shift towards humanising of antibodies, monoclonals, and this reduces the risk of inflammatory immunogenic activity in the biologic. So you can tell, so one of the things about teaching this topic is challenging is just the names of some of these biologics and these monoclonal antibodies. But one of the ways you can help yourself learn the names of these is to realise that the suffix, the last half of the name of some of these products, tells us a bit about the technology. So OMAB is one of murine origin. Zimab tells us that it's a chimeric monoclonal with 65% human sequence. The remainder might be animal. And then all the way through to a UMAB, which is 100% human. Now, like I said, biologics are big dollars. There's enormous amounts of money in these sort of products, and so this is why companies are putting more money into these sorts of therapeutic agents. And if you look at the top-selling drugs of the last 25 years, the 10 top-selling drugs during your lifetime, basically, half of them have been biologics. So there are huge amounts of money to be made by these companies. So if you look at the top seller, Pfizer's Lipitor, atorvastatin, one of the best statin drugs, huge amounts of money for Pfizer, followed hot on the heels by Humira, $75 billion sales since its introduction. And this is a fascinating story because this is produced by a company called AbbVie, and if you follow the news and biotechnology news, you'll know that Humira's patents were expiring, I think it was just a month ago in the United States. It should have expired about 20 years ago, but AbbVie, they're real quite rogues, they kept creating what's called a patent thicket. And they've literally put hundreds of patents on Humira, trying to stop any competitor from making a generic version of Humira. And they've finally all ended all those hundreds of patents, and there's been a real firestorm in courts in the United States addressing this very issue because they wanted to expand their unique production capability as long as they could to profit as much as they can. Okay, so that's the sort of tricks that companies are resorting to try and maximise the benefit, the financial returns on their technology. Okay, you can see a lot of these effective drugs are used for, biologics are used for treating inflammatory diseases such as rheumatoid arthritis. If we look at the current top-selling drugs in the world, or at least in Europe and the US, again, right at the top there, we see Humira from AbbVie. Huge billions of dollar products on an annual basis. So, and again, a whole bunch of these are used in the treatment of rheumatoid arthritis and things like psoriasis. If you look at the impact this is having in Australia, where we probably have a bit more of a regulated market with regards to the use of biologics, but it still hasn't prevented these things from taking a significant toll on our healthcare system. So just in a single financial year, this VEGF receptor decoy costs the Australian government, close to $500 million just for one treatment for anti-cancer biologics. So some of these are, a lot of these are anti-cancer biologics, and then we come down here, these are for treating rheumatoid arthritis. But you can see the costs associated with this. And in the United States, I was reading about a lady that was, you get your health coverage, your medical insurance comes from your employer usually. And then they're interviewing a lady who had psoriasis, serious form of plaque psoriasis, had best years of her life with a biologic treatment, but when she retired, she couldn't afford that biologic anymore, so the psoriasis returned. That's the sort of issues that are being faced in the United States. Here in Australia, the government tends to cover the costs of certain biologics through the PBS, but of course there's a lot of political intrigue and pressure put on the government to put certain biologics on the PBS and other pharmaceuticals. So and it just transfers the cost from the private insurance sector to the government, and that comes with its own implications for our healthcare budget. Now, just a couple of issues to deal with before I wrap up then is that's distinctive to the biologics field is this issue of what do we do when the patent finally expires on a biologic and a company in the other side of the world in a low-cost environment wants to start marketing a product. So rather than call them a generic, we call them a biosimilar. So biosimilar is a non-innovator biologic that is highly similar to and has no clinically significant differences from an approved innovator biologic. Okay, but confirming that a molecule really is a biosimilar and a clinically equivalent copy of an innovator biologic is actually not a trivial thing to confirm, partly because these are large complex molecules. So the chemical analysis of a particular biologic is going to be challenging. It's not just like a mass spectrometry or NMR that any old organic chemist could interpret usually. In a small molecule, these are complex molecules. And one of the things that makes it difficult to confirm that a so-called biosimilar, a generic biologic, is the same as a pre-existing innovator biologic is this issue of post-translational modifications on proteins. So you'll be familiar with glycosylation, and this is a big factor for a lot of biologics. How many glycosyl groups are stuck on the protein has a lot to do with its pharmacokinetics and its immunogenicity and its efficacy. And so confirming that a generic biologic, a biosimilar, has exactly the same glycosylation pattern as a innovator biologic is not a trivial question analytically. But the same holds true for some of these other post-translational modifications that proteins sustain, disulfide bonds and all these other potential modifications. Okay, so in the case of small molecules, pretty easy to confirm that a generic has equivalent molecular identity and so-called bioequivalence, oral availability, same as a original pharmaceutical that you're wanting to copy as a generic manufacturer. A lot tougher to confirm this for a biosimilar producer, and this is one of the reasons why this area is such a minefield. And also adding on top of that too, just think about the fact that we know so much more about how to do cell culture and make proteins than we did, say, 30 years ago. So there's this process of modernisation. So should a company be allowed to modernise its production process for a biologic, even if we know that's going to cause changes in the biologic, so that it's not exactly identical to the one that was approved 20 years ago. So this is a challenging reality for governments to deal with. So today's biologic from the same company might not actually be identical to the one that patients might have got a decade or two ago. So this is a really challenging area. And when is a biosimilar similar enough is a tough issue. People who promote these technologies tell us that the impact that biologics are going to reduce costs over the long term by making people healthier and reducing problems of side effects from small molecules. And some of the projections show these rosy scenarios with decreasing health expenditure across the sector as a result of biosimilars availability. So by approving a biosimilar produced from a low production cost company, maybe there will be benefits to us all. Another term to be familiar with is a bio-better. A bio-better is when a biologic is intentionally improved to improve its, say, its pharmacokinetics and the safety or other issues. So it's a little bit different to a biosimilar. A bio-better is one that's deliberately been altered in some way to make it a better biologic. Okay, that's probably enough for one lecture. I hope I haven't overwhelmed you with all these weird terms, but hopefully it'll just show that there are different ways of treating disease than just giving small molecules to individuals. But they do come with a lot of complicated issues and high costs, at least initially. And so there's a lot of issues surrounding their use. But okay, thank you very much, and thanks for coming today.
You said that you think like the COVID vaccines were pushed too quickly. Is that right? You said that you think the COVID vaccines were pushed on too quickly, right? That's like they are biologics, aren't they?
They are biologics in a way.
Yeah. mRNA vaccines. Is that
So
right?
they've modified one of the 4 bases in RNA to improve the stability, because RNA is very stable, it's working very rapidly, so they put in uridine in there to improve the stability in the cell, so you get more copies made. I think it's fairly controversial technology, not really. I've got trigeminal neuropathy 30 minutes after Pfizer lost
seriously?
sensation.
Yeah.
They do have neurological.
So you think that is that the main side effect, or is there other? I thought there was side effects as well. Is that the only side effect? Or I thought there was.
I did have cardiovascular issues.
Lots of people having cardiomyopathies, things like that. Yeah. How did they get approved? Was that just the FDA was the sort of the rushed?
Yeah, I think they came to different people, governments.
I reckon in 10, 15 years from now, the government website, did you get the COVID vaccine? You may be entitled to compensation.
I think so. I think,
Yeah.
I think because it's 10 years' time, 20 years' time, we'll really look back at this period and say, what did we do?
Because some of them are quite, the potential for toxicity is quite high.
Yeah.
Especially with the spike proteins and things like that.
Yeah. But also the inherent formulation of them, because nucleic acids have a strong negative charge on the sugar phosphate factors.
Yes.
So not only their size makes it hard to get them into a cell, but negative charge also. So they've got to be neutral. So that's why they formulate them in these special synthetic lipids that have a positive charge on them to counteract the negative. But that's non-natural.
Yeah.
So they have toxicity issues. I think that's what you'd want.
Seriously. How did you recover from that? Was it just you just got over it? Or?
So I've had no, still good.
Seriously?
19 months ago.
You
18 months. Yeah, 19 months.
still there?
Yeah.
Wow. What great. Which one was that? Was that Pfizer?
Pfizer. Yeah, and they don't publish much. Normally, drug companies will publish the science based on their products.
I mean, they just got so rich from this as well. Like Pfizer, they've just made so much money.
Yeah.
So much money. Like they would have just been saying, yes, thank you.
Yeah, exactly.
It's billions, because it was mandatory. Like we didn't have a choice. Yeah, it's crazy. Yeah, I regret it.
I won't let myself have another mRNA vaccine for a while.
No,
That's
absolutely.
all some of these problems.
I mean, the clinical trial process for normal vaccines is 10 years, right? Usually in excess of a decade. What was it, 18 months? They pumped this through?
Yeah.
What toxicity models did they use that they thought was an adequate representation of?
Yeah, exactly.
I mean, how?
Yeah.
No.
I mean,
Yeah,
if you want to do something like that in the vulnerable, well, if they were old folks that really vulnerable to COVID, that would have been fine, I think.
The vaccines did
For everyone?
them badly as well. Like a lot of elderly people had some really bad cardiac events from the vaccines. Especially the elderly people. But cardiomyopathies, I think for AstraZeneca,